study published in the Journal of Shellfish Research. Since May 2006, abalone viral ganglioneuritis caused by abalone herpes virus has spread along the coast of Victoria and at times devastated wild abalone population, causing high mortality up to 90% in some areas. However, some abalone from the affected populations survived the disease and thus may be naturally resistant to the disease. So how do some abalone survive AVG and some don't? What makes the disease pop up and where does it originate from? I mean, maybe it's just me, but as we learn more and more about the AVG disease, well, I just want to know more. I want to know why and how it could have popped up here in South Australia and what can be done about it, if anything, if there are any cures. Because in this next half hour, we're going to check in with a researcher who's been working towards the development of antiviral therapeutics against the disease for our abalone. We'll hear what that contains in this next half hour. Now we know abalone viral ganglioneuritis is in the region. We know it's been found off the coast in one reef area off Port MacDonnell, and there's testing underway now to determine if it's already spread elsewhere. There's a control area in place now stretching from Nene Valley all the way to the South Australian-Victorian border, where all fishing activity is currently prohibited. You cannot fish, bait, spearfish, dive for abalone or take from the control area in any way, shape or form, so as to limit any potential spread of AVG. But now it's here. Is there anything we can do other than just letting the disease run its course, which seems to be what we're hearing from others on the topic? Well, researchers at the La Trobe University are currently working on the development of antiviral therapeutics against the AVG in Australian abalone. Jacinta Agius is the PhD candidate with the university who's working on this development. Jacinta, good morning. Thank you so much for your time.
Good morning. Thank you so much for having me.
Now let's take a step back. What can you tell us about where AVG has come from, where it was first cited in the world?
That's a really great question. So AVG has been cited in many different countries around the world. So it's been found in Taiwan and in China. But, more recently it's come to Australia where it's caused quite a few outbreaks in Victoria, in Tassie, and now in South Australia.
It's been described to us as almost like a bit of a cold sore. What is it? Is it like a type of herpes?
Yeah, so the actual pathogen that causes AVG, which is the disease, is called halotid herpes virus. So it is a herpes virus. So we hypothesize that due to it being a herpes virus in its nature, it does have that potential ability to lay dormant within the abalone until a major stressor event happens, and then that's when the abalone gets sick. So similar to humans, when you are infected with herpes simplex virus or HSV, you carry the virus, you're fine most days, but then something stressful happens, and then that's when you have the cold sore symptom pop up.
So abalone populations could already have it, it just could be dormant, but to get that, they have to be infected with it.
Yes, exactly right.
Right. And what would be a stressor event that could cause it to become active?
Yeah, so we still need to do a lot more research in this area to be sure of what is happening here, because our current understanding of HHV1 or AVG dynamics is limited, but drastic weather changes, so huge swells in the ocean or temperature changes may potentially, cause AVG to, pop out and abalone start showing these symptoms of AVG.
And Jacinta, how does it spread? Do reefs need to be touching for abalone to spread it? I mean, how can abalone make long distances to other reefs?
Yeah, so that's also a really great question, and it's still something that we don't exactly know. So it's still our information on this is still very limited, but it is a potential way in which the abalone are spreading the virus is through their mucus, through the water column. When we conduct our AVG trials within our lab, we can see that HAHV1, so the virus, is present within the abalone's mucus. So that could be one of the ways that the virus is potentially spreading from abalone to abalone, but we still need to do a lot more research in this area.
Wow, that's so interesting. Is it something that we've seen in other marine life? Is this quite rare for a marine life to have a disease that can lie dormant?
So that is a really, really good question as well. So the closest known relative to AVG is actually another herpes virus that infects oysters. And those two viruses are the only two known herpes viruses present in all invertebrates, so all organisms that don't have a spine.
Wow.
So it is quite, in its own way, a special little virus.
Quite rare then in the animal kingdom.
Yeah.
Now, I've been doing research on this and I've been really interested to hear examples of when AVG is found in abalone colonies, reefs, but then some of them survive and they're able to grow resistance.
Yeah, so this work is not something that's specifically done by my group, but it is what we've been able to show is that within our group, that if you basically immune prime abalone, which means basically setting them up. Sorry, give me a sec.
No, it's hard to explain. So do they?
Yeah.
Does the abalone need to have something done to it to make it immune to it, or?
Yeah. So within my particular project, what we've been able to show is that immune priming abalone prior to virus infection, which basically means injecting them with an immune primer, which can be described as something that is like a non-infectious part of the virus, in a sense.
Like a vaccine?
Like a vaccine, yeah. Is able to protect these abalone from subsequent HAHV1 infection.
Right. And is this being used in Australia?
At the moment, not at the moment. So we're still working on this strategy because the work that we've been doing, although this is the work we've been doing has been able to show that we can protect abalone this way, it's not a solution that is feasible for larger aquaculture facilities. So that is what our recent project is looking at now. So how we can take this strategy and make it a more large-scale approach for aquaculture facilities and such. So the way in which we're aiming to do this is by making a synthetic immune primer. So remembering that's like a non-infectious part of the virus, and utilising this in immersion trials and feeding trials. So essentially bathing a lot of abalone in the immune primer or feeding the primer through food and then virally challenging the abalone to see if they can be protected that way.
Would that then work the same way a vaccine would? They might still get AVG, but they wouldn't, the mortality rate would be less.
That's also a great question and something that we still need to work on and need to understand. So our knowledge in this field is really quite limited. So we, I guess, are doing what we can to for the industry with the knowledge that we have right now.
Yeah, but you explained just then how rare of a thing it is to see in invertebrates. So very interesting, and you would have to take every step and every hypothesis and every test to make sure you're doing the right thing.
Yeah.
Jacinta, is there any other approach to stopping the spread of AVG once it's in a reef? I mean, I was reading through a Department of Agriculture flowchart which recommends perhaps emergency harvesting if only a small amount of AVG infected abalone is detected. Does that stop the spread, or could it still just pop up in other abalone, by themselves in that sort of dormant herpes type virus?
Yeah, so I guess like an immediate approach, such as, setting up the, control sites where, you can't fish and things like that is probably the best way for the moment to stop the spread. But as you mentioned before, as I've talked about, due to this virus being a herpes virus, there's no way to really know or at the moment, there's no real way to know if abalone are infected with the dormantly infected until, a big stressor event occurs. So I guess, that's why we are working so hard and are so passionate to find a solution for this, because with AVG, it's kind of like, not if it's going to happen, but when. It's a bit of a ticking time bomb.
How much of your time and research have you dedicated? How many years have you been researching on this? And how far into the future do you think it is to see a cure or antivirals or a solution for this?
Yeah, so myself personally and my group have been working on a solution for AVG for about 4 years now. But more recently, this massive project that we're working on now, is funded by Fisheries Research Development Corporation and the Australian Abalone Growers Association, and we, are all collaboratively trying to, find a solution. But science is science, and it takes time. It takes time. So we're hoping that we'll be able to, have some, good results and solutions come out soon, but it does take time, unfortunately.
Why abalone? I have to ask, of all the things you can study, why abalone?
That's also a really great question. I think I'm just intrigued by them. They're just intriguing. So there are so many different types of abalone, and they're just cool little critters.
They are. They're funny.
And I think, I've been drawn to doing research in this area because Australia really needs it. So, I definitely want to use my skills and expertise, in something that I can see will have direct implications for, our Australian abalone industry. Really important.
Well, Jacinta, good luck on the future studies. We'll be waiting to see when your name is on those viral therapeutics being used finally to help the abalone. Thank you so much for filling us in on this research this morning. A fascinating field of discovery. Thank you.
Thank you for having me.
That is Jacinta Agius. They're a PhD candidate with the La Trobe University who is working on developing these antivirals for abalone. There you go. It is just about to turn 8 o'clock.